Alright, this is my special topic presentation number 2. I chose the topic, why the wing markings are so important. Here's my introduction and question slide. I'm going to go off with the first one. The fly is a sheep in wolf's clothing. My special events topic is essentially about how a fly is relevant to a sheep in wolf's clothing. The tephritid fly, also known as Zonozometa vetigera, does the exact thing people use this metaphor for. Now let's see how this relates to a sheep in wolf's clothing. The wings on Zonozometa vetigera have dark bands, making them very distinctive. The wing banding pattern resembles spider legs. Entomologists noticed that when the flies were being disturbed, they began holding their wings perpendicular to their bodies and waved them in an upward and downward motion, which mimicked one of their predators, the jumping spider. I go to the third bullet point. Mimicry of a predator's behavior by its own prey had never been recorded, which is true. We see reports all the time of how predators mimic their prey to capture their prey easier. But we do not see prey mimicking their predators to escape harm reported very often. However, Eric Green and his colleagues proposed that the fly uses its wing markings and wing waving display to intimidate the jumping spider itself. Once it was seen what the tephritid fly seemed to be doing, the researchers had to dig deeper. This is where the main and most important question was asked. Were these flies doing this to avoid predation from jumping spiders who are fast and have a nasty bite? Since jumping spiders are seen as pretty threatening given the two reasons above, the researchers began to broaden their question, asking if the flies were mimicking the jumping spider to not only fool the jumping spider itself, but also a wide variety of other predators. Both mimicry explanations are plausible hypotheses about the adaptive value of the fly's winging waving display, but in order to see if their hypotheses are true, they must test them. Green and his co-workers set out to do so with an experiment. Here's my experiment slide. By having a successful experiment, they must base their hypothesis on a precise question, in which states, do the wing markings and the wing waving of Zonozometa vetigera mimic the threat displays that jumping spiders use on each other, and thereby allow the flies to escape predation? The scientists came up with multiple hypotheses of this question. In this specific experiment, the scientists came up with 3 different hypotheses explaining why it would seem as if the fly was mimicking the jumping spider. I'm going to go off with the first one. Flies do not mimic jumping spiders. Hypothesis 1. This states that the flies are not mimicking the jumping spiders. This is a potential possibility because other flies have dark wing bands and wing flicking displays that are not used to deter predators. In many other species of flies, those characteristics are used during courtship. The second one, flies mimic jumping spiders, behave like spiders to deter other predators. The second hypothesis poses the possibility that the flies are mimicking the jumping spiders. However, it is meant to deter predators other than spiders. Other predators that target the flies may be intimidated by a jumping spider, or in this case, a jumping spider mimic. These include, but are not limited to, other spiders, assassin bugs, praying mantises, and lizards. The third one states, flies mimic jumping spider to deter jumping spiders themselves. Hypothesis 3 says that the flies mimic the jumping spiders and is meant specifically to deter the jumping spiders themselves. Now let's take a look at this diagram. In order to put all 3 hypotheses to the test, they needed flies with some of the parts of a tephritid fly, but not all of them. When configuring possible ideas, the scientists figured out that they could cut the wings off of a fly and proceed to glue them back on with Elmer's glue. They also determined that they could cut the wings off of a tephritid fly and glue the wings of a housefly onto their back. This worked to help to change the variables in the experiment because household flies have clear and unmarked wings, unlike the tephritid flies. Despite the fact that the scientists had cut off and glued on the wings, the tephritid flies still continued to wave their wings and fly in the normal way that they had with their original wings. Using all of the surgical procedures with the wings, the scientists were able to create 5 different experimental groups of flies. The researchers starved 20 different jumping spiders from 11 species for 2 days before the experiment, then presented the spiders to each of the 5 experimental fly types in random orders. The results were pretty clear right away. The jumping spiders retreated when posed with a fly that gave the wing-waving display with the marked wings, yet attacked the flies that lacked the wing markings, wavings, or both. This is my central experiment results. The spar is used to show the results and how each of the jumping spiders reacted to the 5 experimental flies. A majority of the flies that waved marked wings remained alive, while the flies with no markings or wavings were attacked and even killed by the jumping spiders. Looking at Group A at the top, the unaltered Zonozometa flies shows that out of the 20 jumping spiders, 15 of them retreated from their test flies in Graph A, and 5 of the spiders killed their test flies shown in Graph C. Graph B has the Zonozometa flies with their wings cut and re-glued had 15 of the jumping spiders retreated from their flies. 2 had stalked and attacked but did not kill their flies, and 3 killed their flies. I think it's safe to say that most of the flies that waved marked wings in which are groups A and B survived their encounters with spiders and scath. However, the not so lucky ones were the flies that lacked markings or waving or both were attacked, and many of them were also killed. Here's my conclusion. The defining and testing of the effective control groups were critical in the experiment. In this specific study, groups A and B in this chart seen before served as the control groups. These were the controls because they showed that cutting off and gluing the wing spike on the temperature flies had no effect on the reaction of the jumping spiders. This showed that when the temperature fly in Group C was attacked, it was because it no longer had the markings on its wings, not because of the wing surgery. All of the controls and experiments must be treated and handled exactly the same. It was absolutely essential that Green had used the same test area, the same time frame, and the same word usage of predator response in each test, using the same condition every single time with each variable less a research to avoid bias and increase the precision of the data. Overall, Green's experimental design was successful due to making sure the experiments were replicated for each one. His experiment allowed independent testing of the effect that predator type, wing type, and wing display have on the ability of Zonozometa flies to escape predation. Green's experiment allowed us to state that hypothesis 3 was proved to be correct, that temperature flies do in fact mimic the functions of the jumping spider to deter predation by the jumping spider themselves. And here are my references.